Adsorption-desorption chromatography. In adsorption chromatography, the molecules are either bound or not bound to a solid surface, also known as the binding mode. A column is equilibrated, meaning the solid phase is conditioned by passing a solution through it that resembles the pH and ionic strength of the product containing solution. The column is then loaded with the product containing solution, where it adsorbs to the functional surfaces of the stationary phase. Impurities will also bind. However, they can be removed by a washing step, which involves washing the column with buffer. The washing buffer is strong enough to disrupt the interactions between the stationary phase and the non-target protein, yet leave the adsorbed target protein in place. The target protein is then eluted by using a different buffer, or by changing the properties of the buffer to disrupt the binding of the target protein to the stationary phase. The separation of the target protein from the impurities is possible because the components behave differently in the elution and washing buffers. The properties of the buffers, such as pH or salt concentration, can be altered to achieve selective desorption from the stationary phase. Often, the washing and eluate is collected in several fractions. At the end of the process, the column is washed in preparation for the next batch or for storage. Size exclusion chromatography, SEC. SEC separates proteins based on the molecular size, molecular weight, and, in certain cases, the shape. The stationary phase or matrix consists of beads that contain pores or cavities, through which small molecules can pass, but larger molecules cannot. This type of separation works because proteins are distributed between the volume of the solution inside the beads and the volume of the solution outside the beads. As they enter the pores of the stationary phase, small proteins are slowed down in their transport through the column. The larger molecules that are prevented from entering the pores of the beads continue in the fluid stream. Medium-sized molecules spend an intermediate time within the pores of the beads. They can be fractionated from large proteins that cannot access the beads, and from small molecules that spend most of the time within the beads. Size exclusion chromatography is also called gel permeation chromatography or gel filtration. It involves packing a column with a gel comprised of uniform beads containing pores of a narrow pore size distribution.